After this, what method we use for and why we use this method? SNMNT, non-synonymous multiple nucleotide polymorphism. As I told you before that we will discuss another letter, MNP. Okay, MNP means multiple nucleotide polymorphism. When there is change in the same position of two or more nucleotides, it's called multiple nucleotide polymorphism. And also in our mutation, there's a change in 220 and 221 location. The first nucleotide was G and A. G and A changed to TT, means insertion deletion. TT insert and delete GA. The result of this mutation is glutamic acid change into leucine, and 74 means position 74. First amino acid was glutamic acid, and the mutation amino acid is leucine. And also this is confirmed and Patke et al. 16. The confirmation, we do confirmation in Ensemble Genome Browser. This was reported in the Ensemble Genome Browser. For confirmation, we do the search in Ensemble, either this type of mutation can possible, is it is valid mutation or not. So for FASTA sequence, we use UniProt and extension number is KM7RTU4. And on the right side, we have the overview flowchart of our project. First of all, we point out the non-synonymous multiple nucleotide polymorphism from literature. We read article and we point out the mutation that was novel mutation and the novel gene, and also we do confirmation and Ensemble Genome Browser 97, and also we check the pathogenicity of this gene and the subtools of single nucleotide polymorphism, SNP tools, and also we build 3D structure. We predict 3D structure and Raptor X. Raptor X have ability to produce, to predict, to create, and 2D structure and 3D structure. And also further we do mutagenesis and superimposition and 3D structure comparison and PMOL. For protein stability, I did I-mutant2 and Phd waiver. So result. Pathogenicity result, first of all, we input sequence and polyphen 2 with the position of mutation, and also with the current mutation, and also with the old amino acid, and also with the new amino acid. So it gave us the functional prediction as damaging. Means this mutation has some damaging property. Okay, the score of damaging is 0.99. It means near to 1 at effect have some functional property. And also we check the sequence alignment, multiple sequence alignment. The alignment was highly conserved either side of our mutated location. For second, we check the mutation and second tool, which is SIFT, and also the function prediction, protein function prediction is affecting protein function. So till give us the predicted result is this mutation can affect the function of protein, and also it gives us predicted score values. Means some tools contain 5 to 10, some tools contain 1 to 10, and also there will be the range of damaging, a range of normal, a range of neutral, and a range of pathogenicity. So Swift gave us a predicted result 0. 0 means damaging, and also it gave us the prediction of sequence conservation. It is highly conserved. So this is the result of Philippine 2. We can see it also contains the probably damaging, means this have the ability to damage the function of protein. And also if we can check the prediction of multiple sequence alignment, and our query with the other, it is highly conserved either side of our. If we check the location of our gene, there is some non-conservation part, means our gene shows that the mutation part is non-conserved because there's occur mutation. So the wild type of amino acid was conserved, and now the mutant type of amino acid is non-conserved. If we check the result of Swift, so it also leucine is substituted, glutamic acid substituted into position of leucine, and also substitution into leucine and other amino acids we can check. So the prediction result of Swift is affected protein function, and also the position is E74. If we check the E leucine position, which is 0, it means affect the function of protein. And also we check the prediction of mutation and ProVen. What gave ProVen function prediction is deleterious, and also ProVen score is -8, and cutoff value is -2.5. These are statistical analysis that are automatic result of the prediction result of the tools, and also it depends upon SVM. It depends upon the simple vector machine. It is a type of algorithm that give us the statistical analysis of the mutation, and also we do the prediction on photon, which tell us this is damaging, and also the score of photon is -6.8. And also there is different other parameter: damaging, neutral, pathogenicity, deleterious. So ProVen, this is the result of ProVen. And also FATM gave two types of results. First, FATM tell us the predicted result of the mutation, and also if the mutation is concerned with other disease, may concerned with other disease, this also gave us. So FATM gave us the damaging result, and also for further information, for more information, it gave us that this ability, this disorder, this abnormality may be some function malformation, some changes, some disorder in head, face, skull, ear, like that. So Phd-SNP result. Phd gave us neutral result. The reliability index is 4. So this is the result of Phd-SNP that gave us neutral, and the prediction index is 4. It means that we will not focus on the Phd-SNP because other 5 different tools give us deleterious, damaging, pathogenicity of the prediction results. So we will check our own statistical analysis that out of 6, here only 5 tools give us the pathogenicity result of this gene is this mutation is deleterious, damaging. So for secondary structure analysis, as I told you that I use PSI PRED and Raptor X. Raptor X result. This gene contain helix, and also this gene contain alpha helix, beta sheet, and coil. So alpha helix of this gene is 22%, beta sheet is 0%, and coiled is 77%. And both wild type and mutant type. What mean by wild type? Wild type means the accurate formation. That means the real, the functional protein. Mutant means abnormal protein. And also we check the solvent accessibility of both wild type and mutant type. Solvent accessibility of wild type is 85% exposed, 9% median, while 4% buried. Means what will be the effect if this gene come in the tissue or if this gene come in the skin. There are some exposed region of this gene outside of the skin, and there means the formation of this gene like inside the cellular functional, 9% is 85% exposed, 9% is in median, and 4% is buried. While the mutant type of protein, 85% exposed, 8% is medium, while 5% is buried. And also, what are the formation of normal glutamic acid? Normal glutamic acid have 5.7 alpha helix, 0.1% beta sheet, and 4.2% coil. Substituted leucine. Glutamic acid changed to leucine that contain 96.8% alpha helix, 0.1% beta sheet, and 3% is coil. The solvent stability of normal glutamic acid is 39.9% buried, 34.9% median, and 25.2% exposed. The solvent stability of mutant leucine. So now we are discussing with abnormal leucine, abnormal protein, and mostly we are targeting to leucine. 48.1% is buried, 32.3% is median, and 19.6% is exposed. If we look the data, the statistical analysis, the data of helix beta sheet, there are some minor change and some major changes. And also we are looking to PSI PRED results. PSI PRED gave us only secondary structure. The secondary structure of PSI PRED is 2 helix and 1 loop. It means this gene contains 2 helix and 1 loop, and the mutation is in first helix. So this is the Raptor X result. If I give zoom to this result, leucine 74. This is leucine 74. This and 6.8 helix, 0.1 beta sheet, and 3 coil. And also it is the secondary structure. As we are looking about the solvent stability, which is 48.1% buried and 32.3% median and 90.6% exposed. If we look to the PSI PRED result, so it contains 2 helix. It gave us many different color. So the color depend upon the secondary structure, or we can say secondary structure depend upon the different color. If some color appear inside the secondary structure, the amino acid will be formed this type of formation and say the secondary structure. Yellow strand, we are looking to helix, which is pink color, and someone is looking for extracellular activity of amino acid, that will be orange color, and someone is looking for the disorder or protein binding site, which will be white, and different colors show different formation and secondary structure. So 3D structure modeling of mutant protein we do in Raptor X. And Raptor X, I did 3D modeling by ab initio method. So first of all, we know about ab initio method. Ab initio is a type of method that amino acids are joining one by one and it form a structure. Why I did this? I did mostly Swiss model and also IHSR and also modular. It gave me a little bit result about 3D model because prediction depends upon a template. So there were a lot of template of this gene, but our mutation were not present in the predicted model of 3D structure of this gene. When I did prediction of 3D structure, 3D structure in Swiss model, so my mutation location is 74, it was disappear. So I was worried that I am working with 74, it's not present in the prediction model of 3D structure and Swiss model. I use modular, it depends upon template. So I read about the Raptor X. So I did this because it gave me complete structure, complete 3D structure and predicted form without any template. It gave me an evaluation method, how amino acids come together and how they build a structure. So this is the mutant type structure. The red point shows the mutation. And also I visualize this structure and symbol. It is the visualization tool. We can see some tools because we can do further analysis in it. And also the structure of this gene BHLHA9 was not determined. All determined structures are present in PDB, protein data bank. So I searched at protein data bank, there was no determined structure. So that's why I create a 3D predicted model of this gene. So I also did the mutagenesis of PE74L. E means glutamic acid, 74 is the position of amino acid where glutamic acid change into leucine. And also I do mutagenesis and I found 4 different rotamers. Rotamer means the different position of amino acid inside the 3D structure. So the best rotamer was 60.5%, means greater change occur due to this position, this angles of the gene, of the amino acid. So other rotamer were 34.1%, 2.1%, and 2%. And also we can say high percentage edges percentage equal to low clashes. And also this is the 3D structure of wild type. It is located in the green position, and the muted 3D structure is and mutation point is in red. And also the position of E74 of glutamic acid and picture C, and also D picture position of leucine at 74 is also present here. So we can continue with that. How we do superimposition? What is the need of superimposition? Actually, superimposition is the way to compare wild type structure and mutant type structure. I did the comparison between two structures and I saw that there are some changes inside the structure due to rotamer formation, high rotamer formation, and also the location, that means the size of the amino acid, both amino acid are different in this disorder. Due to this, the size of leucine, large size of leucine, it may bind with other amino acid. It may bind with uh, other protein. Due to this binding formation, due to this reason, it can affect and give the abnormal protein formation. And also this mutation is an active site. 74, 71 up to 78 were the active amino acid of this gene, active site amino acid of this gene, of this protein. So 74 is already involved in the active site. Due to this mutation, it gave the abnormal protein formation to this gene. And also we calculate RMSD value, which is 0.034. It is not spellingly good. And also prediction of protein structure stability. I did protein structure stability and I mutant and CISPD behavior. The difference is too much high. And also decrease in stability, which is 0.7 kilopascal per mole, kilocalorie per mole. And also CISPD behavior giving a huge amount, kilojoule per mole. If I calculate the difference between the two values of I mutant and CISPD behavior, it gives me a lot of high, huge amount of value. It means that the protein structure or the mutation has some effect on the whole protein structure. And also discussion, BHLH mutation falls in three categories. First one, the encoded protein is transcription factor involved in red development. And also mutation is that this gene has been associated with syndactyly, major arithmetic syndactyly, with the phalange reduction and camptodactyly. And also copy number of variation of locus head. This gene has been linked to the split hand and foot malformation with long bone deficiency. So why I did discussion like that? Because there was no bioinformatics and silico analysis did before when I was doing this. So I just like that I don't there was no work so that I compare my work with them and I suggest a discussion that my result is this, the given result of pupil is this, that are worked before me. And also conclusion, we conclude that non-synonymous multiple nucleotide polymorphism has functional impact on protein. And also non-synonymous multiple nucleotide polymorphism has structure impact on protein. And also protein function is greatly influenced by 3D structure of protein. And also I did this based up in silico evidence. The mesoexocytic syndactyly was with phalangeal reduction and polydactyly complex is mainly a split hand foot malformation with long bony deficiency. Future recommendation for the project is screen all the mutation and BHLHA9 gene held that. Up to now, screen all the mutation and check the effect, check the in silico SNP analysis, and do some bioinformatics structure function analysis, and also do MD simulation, molecular dynamics simulation in it, and also determine a well-validated structure. And also for further functional correction, characterization of mutation is needed for confirmation of the hypothesis. And also annotate the non-synonymous multimonad nucleotide polymorphism that falls in the fold disturbance category, and also give comprehensive in silico evidence of integrating selectively and non-covalently with any of the basically loop helix superfamily of transcription factor and pattern regulatory components as transcriptional network of mini development pathway. It means that we give comprehensive analysis of basic of this gene and also super family that are functional like this gene. I mean the family that are involved in transcription factor and also those family that are involved in the regulatory component and also at this gene developmental pathway. There will be other genes that will be involved in the developmental pathway. So it means integrating all the other genes that have similarity, that have ability like BHLHA9. So acknowledgement, I thankful to my head of department and my supervisor who helped me, and also other teacher, other professor that helped me in my career-based studies, and also other teacher that teach me in my bachelor degree. And of course, I must thank to my project mate that give me motivation and support either at financial or emotional. So thank you for your attention.
Okay, thank you, Abdul, for your wonderful presentation. So if anybody has any kind of question, then Mr. Abdul is ready to answer.
Okay, I have a question.
Okay.
How did you validate that 3D structure?
What?
How did you validate that 3D structure?
I did only in Galaxy web server.
What? And can I know about the energy minimization from Swiss PDB viewer?
Yes, you can check the energy minimization and Swiss PDB viewer, and also if you want to check the stability of your protein, that will be an iMutant server. These are actually servers that you have to download, and you have to put your protein sequence, and you will check the energy minimization and all things.
So you used iMutant
For stability. As
okay,
I want to check, yes, I want to check that my protein was stable or not. Because stability, I predict the structure to Raptor X. It is the Ab initio method, that how different amino acids come together and what type of structure they create. So due to this reason, first step will be stability, and then will be energy minimization, and then will be validation. For validation, there are different tools. You can check one by one.
Okay, thank you.
Welcome.
Hi, Abdul. Thanks for your kind presentation. So there was some disturbance in the internet. So there are few questions that are from the theory section that I could not understand due to the with disturbance in the internet connection. And there are a few questions from the experimental part also. So if you can address those things.
Okay.
So the first thing is in the beginning you mentioned that in the LHH domain, it can form homodimers or heterodimers. So heterodimers, it forms heterodimers with what other protein?
Yes, this protein, this BHL gene have two type of domain. One is basic helix domain, other is basic helix loop helix domain. One can bind with one produce EBOX. EBOX is hexadimer protein nucleotide sequence. Hexadimer means 6 proteins that are involved in the transcription site. Okay, means regulatory protein. It is the section of the regulation that the regulation will start of other genes when this gene is bind with other protein or with other gene. It means that the hexal nucleotide means that the regulation of other gene is dependent upon BHLHA9. Okay, this is the first domain. Second domain is produce homodimers or heterodimers. Homodimer means the sequence that have same. It is the special sequence that has same number of the same nucleotide. We can say that ATG, ATG, ATG. These are the homodimers. ATG comes 3 times. Okay. Heterodimer means ATG, TTG, TAA, like that. Means the formation of different nucleotides come together at form heterodimer, and the formation of same nucleotide is called homodimer.
Okay, that thing is clear. And then you mentioned something about the mutation at the active site. So how did you select this particular mutation, as well as what is the role of that particular active site other than binding to that nucleotide sequence?
Okay. So I read this in the literature. This mutation is an active site. Okay. So I confirmed this mutation from Enzyme Genome Browser. All the accepted, validated, and determined mutation are submitted there. So I look the mutation that I read that this mutation is active site and literature review, and I suggest that if I work in this mutation, there will be some changes in the active site, and I examine the changes like that the position and the size of glutamic acid is small, then the size of leucine amino acid. If we compare the size of the leucine and glutamic acid, that have a huge amount of change. It can disturb the other amino acid side amino acid, and also it can bind with the other chain of whole of itself amino acid of itself 3D structure of itself protein.
Okay, and by active site, what exactly is the active site doing other than binding to that that nucleotide domain?
Okay, so as I discussed, that this protein helps in the development of limb development. So if there is a active site function is to perform something functional function. I mean that if there's a lock and key model, actually we can apply here lock and key model. If you have difficulty to open a lock, it means that your key is not fitted in the lock. Okay, so when this protein do function, the ligand, the active molecule bind with active site and trigger the function of this protein. And also in sense that some protein functional combinedly. If there is sorry, if there is change in active site, so the function will be disturbed.
Okay, I'm still not very much clear about what exactly the active site is performing. Suppose some proteins have protease activity, some have glycosylase activity. In that context, I was asking what exactly is the active site doing over here? What kind of catalysis is this active site doing?
it triggers the function of protein. Actually, active site have ability to trigger the function of protein. And also mostly if we look, mostly active site ligand bind with active site protein. Ligand means active molecule, the key of the active site that are bind and perform a specific function, perform a target function. If there is no ligand binding with this protein, definitely this protein will be abnormal, and there will be malformation of digit or limb development.
Okay. So in some of the slides, you mentioned that it has a conserved region with, so conservation with respect to what? With respect to other isoforms of this gene, or with respect to the same protein in different organisms?
A different protein with the same protein with different species of different organism, conserved region.
Okay. And what about the different, does this protein has different isoforms in human also?
No, they have other isoform in human. I just the prediction result was that this gene have other other we can say that other functional conserved region of this gene is found in this species, this species, this gene, and also the accession number are mentioned in the result.
Mm-hmm. So
I will go to the slide. I will go to that slide. Let
Mhm.
me give me 1 minute. Sorry. Okay. This one you want to discuss.
right.
Okay. So if you look to my query, there are some regions that are highly conserved. Okay,
Mhm.
means from the top, GGG are all conserved region, but I haven't put the exact location of my mutation where the mutation occur. The G change into L, E change into L. So on the mutation area, all my gene was conserved at the other side of mutation, but the position where mutation occur was not conserved. Means the change of amino acid G to L, E to L, glutamic acid 2 leaves.